Fine. So we are starting with this new lesson called Inheritance and Variations, okay, which is a very important part under common biology or general biology topic that is called genetics. Okay. So this topic of genetics is actually very, very interesting to learn, okay, and not very difficult also. And this is something the discoveries have happened long time back. Okay. And after that, the research in genetics is still going on. That means it has not stopped.
Yes.
Because genetics is as a field which you can say becomes the base for so many other different fields. Okay, that's why it is not completely under plant sciences or only animal sciences or microbial sciences. This is general biology. Okay, applicable for all organisms. That is genetic. Okay, so let us take a look. Before we will start right from the scratch. Okay, and there will be certain key terms, important terms वगैरह आएंगे वो पहले समझना है अच्छे से. Okay, and based on the terms, there will be some genetic crosses and all which we have to learn. And I will make you do some crosses also on the spot. Okay.
Okay. Fine.
Okay. Yeah. So there, but it is very, very interesting. And exam point of view, it is extremely scoring also. Okay.
Oh, okay, fine. Yeah.
So first thing, when you are talking about inheritance and variations, okay, the simplest or general meaning of the word inheritance, it is something called heredity. You know that?
Heredity.
In 9th standard as well as in 10th standard, you had two separate lessons like this. 9th mein you had a lesson called heredity and variation.
Variation.
10th mein you had heredity and evolution.
Correct.
Okay, so heredity is something you have already heard about it. Okay.
Yeah.
So heredity is the other word for inheritance.
Inheritance. Okay.
So basically, what is heredity or inheritance? It's like there are transmission of important information or characters from one generation to the next generation.
The next generation. Yes.
Okay. So the original generation, which is giving birth to the next generation, we use the word parental generation or only parents.
Yes.
Okay. And the parents are the ones with the help of their genes or with the help of their cells. They are transmitting their genes to the young ones.
Yes.
It can be any mode of reproduction. Can be the simplest method like fission also. But there is a transmission of characters from one generation to another. उसको बोलते हैं heredity or inheritance. Okay, the mechanism of how this inheritance takes place is complicated when it comes to sexual reproduction, and it is very simple in the case of asexual reproduction.
Okay, got it.
You recollect something called mitosis and meiosis?
Yes, yes, of course.
There are two main types of cell division.
Yes.
Correct. So in mitosis, one diploid parental cell divides into two diploid parent daughter cells.
Yes.
That is why usko bolte hain equational division. The ploidy number of chromosomes does not change, and it is the same genes which are transferred.
Yes.
Okay. Then meiosis is also called reductional division.
Reductional division.
Yes, because one diploid parental cell divides and gives rise to four daughter cells. All four are haploid.
Yeah.
And those four haploid cells are called gametes.
Gametes.
Okay?
Yes.
So when the gametes are produced, when the meiosis takes place, in the first step of meiosis, jisko meiosis 1 bolte hain, in that stage, there is a very important evolutionary mechanism taking place, which is called recombination, also called crossing over.
Crossing over, yes.
So there is an exchange of genes that is taking place.
Yes.
Shuffling of the genes. So because of which, whenever sexual reproduction takes place, gametes are produced. The next generation which is there, it is receiving the set of genes from both the parents.
Correct.
That's why any offspring produced as a result of sexual reproduction is having characters from both the parents. That
Yes.
is the type of inheritance in sexually reproducing organisms.
Yes.
But in the case of asexual
Sexual
reproduction,
difference.
only one parent is there.
Yes.
So there is no recombination, no meiosis, no variations created. Variations matlab differences in the genes.
Yes.
Getting it? Okay. The diversity which is produced in the genes due to this mixing. Usko variation bolte hain. So there is no variation that is produced in the case of asexual reproduction. Understood these simple facts?
Understood.
Okay.
Yes.
Now,
Yeah.
the thing is, all these points that there are characters which are transmitted from one generation to the other, inheritance, heredity, chromosomes, genes, these were not known to us till the time technology was advanced.
Technology. Oh yes.
See, ये सब समझने के लिए सबसे पहले important क्या है? There has to be the discovery of a cell first. People should be, the person should be aware that there should be a cell in which there is a nucleus, inside which there is chromatin material, which are condensed from the chromosomes. Chromosomes represent the DNA, and the DNA consists of many genes.
Yes.
Correct. Relax. Okay.
Yeah.
So that's how we know about nuclear genes.
Yes.
Okay. And each gene codes for a particular character. And the way it codes for a character is by protein synthesis. You recollect the 10th standard topic?
Protein synthesis. Oh, I remember.
The very first thing I had taught you, protein synthesis.
Yes.
Right? Transcription, translation, translocation.
Yes.
You know, very good.
Remember.
So all that also comes under inheritance.
Inheritance. Okay, got
how the genes are expressing a character. But that inheritance is called the molecular level of inheritance.
Molecular level?
Of inheritance, because the DNA or the genes are expressing in the form of a protein. And
Okay, got it.
Their function. That is molecular. But here we are talking about the genetic level.
Genetic?
We are restricting to the level of chromosomes and genes.
Okay, got it.
So, ये सारा जो studies है, genetics. Here, the beginning of all this started way back in 1822.
1822?
By a person called Gregor Mendel.
I know him. Yes,
You know
Mendel.
him? He was your classmate? Huh?
No.
Gregor Mendel. Okay, he was like actually belonging to a farmer family, a peasant farmer, and he was born in Moravia. There's a place in Austria.
Austria.
Yes, and he was the first one. He was not a scientist. He was what you can say, a monk. Okay, working in a monastery. His father was a peasant. Okay, and he had never used a laboratory, microscope, or anything like that. He was not even aware about the words called chromosomes or DNA or genes or even cell. But he was the one who was the first one to explain something about inheritance without using these words. And that is the most interesting part of it.
How?
Okay, see, what he did was, he had a garden. It was 1822. Life was very simple that time. Correct? He had a garden, and whatever experiments that he did experiments. But out in the field, in the garden. Okay, not laboratory experiments. So what experiments he did was on the pea plant. Sweet pea or the garden pea.
Yes.
Also called Pisum sativum. Remember?
Oh no, sir, no.
Pisum sativum. Okay?
Yes.
Common अपना जो वटाना मटर बोलते हैं, that plant. Okay?
Yes.
He worked on that. And what he did, the reason why he did that was only out of curiosity. He just wanted to know, okay, whatever characters or features are found in a parent organism, how does it go into the next generation? So
Yeah.
in order to
Oh,
answer his own curiosity, he did the experiments and he kept a written record of whatever he did and whatever he got. And that's the reason Gregor Mendel is known as the father of genetics. Okay.
yeah.
See, what he
Yeah.
did was, see, first thing, why Mendel selected only pea plant?
Hmm.
He wanted to observe inheritance from one generation to another, so he could have taken any organism,
Yes.
right? But why out of all the organisms he selected only the pea plant? There are reasons behind it.
Oh,
First reason why he selected pea plant.
yeah.
Reason number one, the pea
Hmm.
plant is easily available. He could have
Okay.
worked on coconut plant also. But
Yes.
imagine putting a coconut seed in under the soil, sowing it, expecting a coconut plant to start. It will wait
Yes.
for the many years to become a tree, again produce coconuts, then he will take that next generation and check. So one Mendel's lifespan would have been less to study the inheritance.
Yes.
And same way for many other plants, which take a lot of time to grow. That's why he wanted such a plant whose generation grows faster and he gets the next generation quickly. So pea plant was one of them. Secondly, second reason why Mendel selected pea plant was it was not very expensive. Okay, and the seeds, the pea seeds, the watana that we consume, they are not very
Yes.
hard seeds. See, उसको अगर हम sow करेंगे तो plants germinate faster, easily.
Right.
These seeds are very hard. A mango का seed imagine. Okay, so they take more time to germinate under such cases. So Mendel did not want all those obstacles. He thought pea plant is convenient. Right?
Yes.
And the most important reason why Mendel selected pea plant, most important reason was that on the pea plant, there are many such characters which are very prominent and very clearly visible.
Characters?
Yes, see this table. There are certain characters on the pea plant which are commonly observed, easy to observe, okay, and can be easily studied. There are 7 different characters which Mendel observed on the pea plant. And each of the characters had 2 contrasting forms.
Contrasting.
See this. First character was the seed shape.
Seed shape.
Pea plant ka seed, uska shape. So some of the seeds, many of the seeds were round, clear round, and some others were wrinkled. So looking at the seed, it can be easily understood. Acha ye round hai aur ye wrinkled hai.
Correct.
So it is not a difficult thing to see.
Yeah.
Secondly, the same seed, uska color. Okay, there were 2 contrasting ones. Yellow with a green with. Again, easy to understand, not confusing.
Yes.
Right. Third was the pod shape. Pod मतलब उसका fruit. मटर जो आप लेते हैं उसके अंदर वो छिलका रहता है ना. I mean the seeds are there. So this is the pod. उसका shape. So some of the pods were inflated. You have to use the same words.
Inflated.
Some of the pods were inflated. मतलब एकदम से फूगा हुआ. And some others were constricted. Can you see the difference?
Constricted? Yes.
एकदम ऐसा wrinkled type.
Okay, got it.
So again, these are characters.
Okay.
Very prominent and easy to observe. And pea plant का और एक advantage क्या है कि in a single pea plant there are many pods and many seeds produced.
Correct.
It comes very fast. Easily the seeds can be collected, sown under the soil, and very soon the next generation also starts. So
Okay.
Possible for Mendel to study many generations like that in a short span of time. तो इतना smartness देखो कि pea plant select किया उसके, right? Then same thing pod का color. अभी seed में देखो dominant was yellow, recessive was green. Pod में dominant is green, recessive is yellow.
Yellow. Okay, correct.
तो वैसे ही याद रखना, ठीक है? Then coming to the flower. Flower
Yes.
color में देखो, dominant ones were purple colored ones.
Correct.
इसमें white colored flowers. तो purple and white को easily देखकर पहचान सकते हैं.
Yes.
Very contrasting.
Yes.
Flower का location. Okay, on the plant, where is the flower produced? So dominant location is the axial. Axial means what? See, if this is the stem.
Yes.
Okay, on the stem, if this is the branch. So if it comes at the junction of the stem and the branch, उसको axial बोलते हैं.
Oh, okay. Oh, the sun.
Okay.
Yes.
And if it comes at the ends of a branch, then it is called terminal. End of the stem or the end of the branch. Then it is called terminal.
Okay, got it.
Can you see that in this picture?
Yes, yes, of course.
Okay. So axial
Yes.
and terminal. So flower का location देखकर पता करने में it was not a very difficult task. Easy to
Yes.
understand.
Right.
And then the most important thing, when the plant has grown completely, what is the height of the plant? Okay, so there are some plants which were growing tall.
Yes.
Some others were growing short. Short को dwarf बोलते हैं.
Yeah.
So you have seen on the plant, the flower, the pod, and the seed. In सबके characters Mendel ने observe किए. There are 7 such characters, each having 2 contrasting forms.
Correct.
So very distinct and very simple, clearly visible ones. And another best part was that out of all these 7 characters, none of the characters were showing a form which was different from these 2. ऐसा नहीं कि सीड के कलर में येलो एंड ग्रीन के अलावा कोई तीसरा कलर दिख गया.
Oh, correct. Yeah.
के अलावा कुछ और दिख गया. Any of the cases that did not happen.
Yeah.
It was either of these 2 only.
Yeah.
So it was convenient for Mendel to study and observe. Right? That's the reason these 7 pairs of contrasting visible characters. There could have been other characters also in the pea plant. But Mendel was restricting to these 7 because they are visible, very easily seen. Maybe there are some characters in the physiology or the biochemistry of the cell and all, but Mendel के लिए वो नहीं possible था. Getting it? These were the ones that Mendel could easily do in the field, in the garden.
Yes.
Okay. So ऐसे करके Mendel had saw this. अभी हम लोग जिसको characters बोल रहे हैं. Now we are aware that all these characters are because of the respective genes. Right? But Mendel was not aware about all that.
Now you can continue.
So I was telling that these terms, that is chromosomes, DNA, okay, characters, heredity, ये सब Mendel को नहीं पता था बिल्कुल, right? So Mendel understood that there is something which is going from one generation to another.
Yes, correct.
Inherited. But that's something which Mendel understood. He could not use a particular word because technically उसको नहीं पता था. Correct? So what? He had used a word called factors.
Factors?
Okay, उसने like उस term क्या कर दिया? He used a word called factors, which today we understand as the word genes. The genes are getting transmitted from one generation to another. Okay, so the Mendelian factors are now known as genes.
Okay, got it.
So based on whatever experiments Mendel did, okay, he because he had restricted to pea plant those 7 characters, right, and certain limitations were there in his experiments. But whatever he did was everything successful. And whatever results he got, a lot of experiments he has done, hybridization experiments and all. Okay,
yes.
And he got really good results. And he recorded all the results in a proper data in a mathematical form. Based on the results that he got, he could generalize something in all the experiments. Okay, there are certain points, certain factors or certain criteria which are fulfilled across every inheritance, जो हर experiment में नजर आए. So he what you can say gave those ideas as principles of
Principal. Yeah. Hereditary.
okay.
Yes.
And later on he just noted it down. See, that time he was not aware that this could be published somewhere or something like that. Nowadays
Correct.
people discover something or invent something, we have an idea, we can publish it somewhere, we can have our own copyright as patents, अपने नाम
Yes.
से वो जाना जाएगा, right? But Mendel के टाइम पे
Yeah.
ऐसा नहीं था, he was not aware, so he just kept a written record. Later on his ideas of heredity were postulated as the fundamental laws of heredity later.
Oh. Okay.
Okay,
Yeah.
वो laws बन गया, and that was done by Correns in 1900.
Coron कौन है अभी?
Correns करके एक और साइंटिस्ट थे, 1900 ईयर में he postulated कि whatever Mendel has put forward as concepts that could be understood as the fundamental laws of heredity. Getting
Okay.
it?
Got it.
Okay.
Getting it.
Now, why was Mendel successful? The major reasons I have told you कि pea plant लिया तो क्यों फायदा हो गया. But his reasons for success involved his careful planning experiments, and he did not do it on one or two plants. He did the experiments on many plants, so large samples.
Oh, okay.
That is also equally important. Whenever this kind of experiments are done, even today scientists always say there should be a large data, a substantial amount of data.
Yeah.
Getting it? That always ensures that results are having the least errors or are without any errors.
Yeah.
Getting it? And
Getting it.
whatever experiments he did on plants, everything he did in the garden, he expressed his results in the form of ratios.
Ratio.
Numerical
Oh,
data मिला.
okay.
Numerical data. Getting it? Contrasting characters
Getting
could be easily understood in his data.
Hmm.
7 different characters that are shown in the table. The best part was that जो Mendel के लिए luck factor था, which Mendel was not aware.
Yes.
That all these 7 characters on the pea plant, these 7 genes responsible for these characters are located on separate chromosomes, and that was a luck factor.
क्या समझा नहीं?
Chromosomes में DNA होता है, DNA में genes होते हैं.
Yes.
And
Correct.
genes are responsible for coding for a character and passing it on.
Correct.
तो pea plant के cell के अंदर जितने chromosomes थे, okay, diploid number was of 14 as the number, so 7 duplicates. So all these 7 characters के genes, they were present on 7 different chromosomes.
What?
Yes. Mendel was not aware about this. Later on it was discovered. But the 7 characters were located on 7 different chromosomes, and that was an important reason. Why? Because if 2 or more genes would have been in the same chromosome, there could have been a possibility of linkage.
Okay, got it.
Linkage मतलब linked genes का मतलब है कि एक ही क्रोमोसोम में 2 या उससे ज्यादा genes and they are inherited together. So in short, for your understanding, अगर ऐसा होता तो it would have been possible that 7 contrasting pairs of characters which Mendel saw, there could have been a third character seen, and then it would have confused कि ऐसे कैसे आ गया. And वो discover नहीं कर पाते.
Yes, got it.
That's the significance. And then Mendel was the one who introduced the concept of dominance and also recessiveness.
Yes.
See those words now. But understood the background behind Mendel's experiments properly.
Understood.
We will come back to the experiments, but before that, let us understand some certain important keywords. Very important words. These words, these terms have to be understood very, very clearly in order to understand Mendel's experiments and even entire genetics. Getting it? तो ये ध्यान से सुन, अच्छे से समझ, ठीक है? So most of these terminologies, ना, we can understand them in the pair, कि मतलब दो एक साथ समझना है. जैसे you can understand character and trait together.
Yes. Okay. Yes. Correct.
Okay, character means what?
Factor.
Any specific feature of an organism.
Hmm.
Can be any feature, morphological, anatomical, physiological, biochemical, anything.
Yes.
But every organism is a combination of these features.
Ah.
Okay, the color of your hair, color of the skin, height of the
Yeah.
individual, shape of the nose, color of the eyes.
Yeah.
They're all characters, they're features.
Yeah.
ठीक है, तो एग्जांपल यहाँ पे दिया, height of a stem is a feature,
Mm-hmm.
color of the
Correct.
flower is a feature, shape
Yes.
of the seed is a feature. So it is
Yeah.
for every organism.
Okay. Got it.
Trait means each character can have more than one possible variant.
Variant.
Yes, for example, listen.
Variety. Oh.
For example, listen, if I say, okay, let's show you like this. See, suppose if I say in human beings,
Hmm.
color of the eye is a character.
Oh. Okay.
So what are the
Yes,
possible
I got it.
colors? What are the possible colors in human beings?
Blue, green, black, brown,
The most common is black,
black.
correct? It can be blue,
Yes.
it can be green,
Yes.
it can be brown,
Yes.
okay?
It can be gray. Yes.
Gray eyes है,
मुझे था बचपन में.
नहीं वो अलग है,
I don't know how.
नहीं, it is black. These are the most common ones.
Yeah.
Now, color of the eye is a character, and black, blue, green, brown are the traits.
Yeah.
So that means an individual or an organism will have one of the traits for a character.
Okay. Got
Now
it.
your eyes are black,
Yes.
so you have black eyes as a trait.
Yeah.
And from childhood, always for the entire life, it will always be black.
Yes.
The color of the eye change every day. सुबह
No.
चलो आज green है, कल blue है. Does it
No.
happen? Of course not.
No.
It is a character which is genetically determined because you are having the gene for black eyes.
Yes.
It doesn't change. Getting it? Okay.
Getting
So,
it. Getting it.
Similarly, एक और example जो easy है और बोल सकते हैं, blood group is a character.
Blood group.
Every
Oh
human
yes.
has got a blood group.
Yes.
Some or the other blood group, right? So blood group अगर character है, तो what are the possible traits?
O plus, O minus,
Plus
A,
minus
B.
नहीं. We say O positive, O negative.
Oh yes, of course.
B positive,
O
B
positive,
negative.
O negative, A, B positive, A, B negative.
Positive,
A
negative.
positive.
See, positive, negative is another character.
Oh.
वो
Yes,
रिसर्च फैक्टर
correct.
बोलते हैं. Okay, so
A, B,
A,
B, O.
B, A, B, and O are the
Yes.
4 possible ones. They are the traits.
Yes.
So what is your blood group?
O plus. Positive.
O positive. So you are a universal donor,
Of course.
right?
Yeah.
So now this O positive blood that you have, is that right from the childhood? Does it
Yes.
change? कि जनवरी
No.
में चेक कराया था B positive था, जुलाई में किया तो O positive हो
No.
गया? No, of course not.
No.
Because a
Yes.
character or a trait is fixed for an individual. Getting it? Height of an
Getting it.
individual. A person can be tall or dwarf.
Yeah.
So genetically, they won't change throughout the life. But sometimes in some characters, in some organisms, environment also plays a role.
Correct.
Okay, environment can play a role. And trait can change. But वो genes नहीं बताते.
Yes.
So understood the difference between character and trait?
Understood.
Okay. One of the traits is inherited and is a detectable variant. That's how you can define it.
Yes.
Okay. Then something called factor, which Mendel had used.
Factor. Yes.
Okay. So factor is what? Yes. Okay. It is a unit of heredity. Okay. It is like a particle or any substance in any organism which goes to the next generation, which is transmitted to the next generation and carries the genes. So in sexually reproducing organisms, the transmission is through gametes.
Gametes. Yeah.
So it is the factor which determines the genetic character of an organism. Remember this point. Okay?
Yeah.
Then
Yes.
similar to that, we have gene. You know what is gene?
Yes.
It is a particular segment of a DNA, a small portion of the DNA.
DNA.
You know the structure of the DNA. Nucleotide,
Yes.
sugar phosphate backbone, nitrogenous base,
Yes.
all that you have learned. We'll study that in even this one, molecular basis of inheritance. Okay. So that particular segment of DNA which is able to express a particular character and also get inherited to the next generation.
Yes.
So
Correct.
okay. Now understand this. Okay. If suppose now genes, when you say, we say it codes for some characters.
Correct.
It's not necessary that there will be always one gene for one character. Okay. Sometimes, most of the time, it is two genes. I'm just denoting a gene like this. Sometimes there can be two genes or maybe two or more than two genes coding for the same character, but which trait to be inherited is decided by their interaction.
Okay, got it.
Interaction between both the genes or more than two genes, whatever is there, and based on that, one of the traits of the character will be determined.
Yeah, got it.
The alleles or allelomorphs. Correct. Also called alleles.
Yes.
Two or more alternative forms of a given gene.
Given gene.
So two or more. It can minimum, it can be two, it can be more.
Got it.
And you are aware about something called homologous chromosomes?
Yes. Homologous, heterologous.
So if there are two such chromosomes which are homologous, that means they are carrying the same genes. We are having a diploid, a double set. So एक maternal chromosome, एक paternal chromosome, right? So suppose there is a gene on the maternal chromosome at this location. So in the corresponding paternal chromosome, the same gene would be there in the same location.
Correct.
Then we call it homologous. Homo means identical or uniform. Logos has come from the word loci, that is position.
Position. Okay, got it.
So the actual word is allelomorph. But in short, we can call them alleles.
Yes.
Understood the meaning of this?
Understood.
Properly, because understand is really very properly, because these are the words which will keep coming again and again when you learn the entire genetics.
Yeah.
Okay. Now, when you say alleles, they are of two types. Okay. There is a dominant allele and there is a recessive allele.
Yes.
Dominant allele matlab, in any combination of the genes, this particular allele will be expressed. Okay, if there is a dominant allele, along with that, if there is a recessive allele, or along with that, if there is another dominant allele, this dominant will be expressed.
Okay, good.
Then it is called dominant. It expresses its trait even in the presence of an alternative allele.
Yes.
Then it is called dominant.
Yes.
And generally, dominant alleles are always expressed more, and those characters are always seen more in a given population. इसलिए उसको dominant करके बोलते हैं. Now, like in this chart, in this table that you saw, how do we decide कि round dominant and wrinkled recessive? Pea plant पे लिखा हुआ तो नहीं है कि I am dominant, I am recessive.
नहीं.
We have decided it. Correct?
Yes.
On what basis do we decide? Because in a population of pea plants, if you see the seeds, you see that majority of the population is having round seeds.
Yes.
Very
Correct.
few are having wrinkled.
Uh,
So under natural conditions, considering everything else favorable and normal, if the ones which are found in abundant
yes.
of the time,
Oh.
that is the dominant one.
Okay, got it.
That is the one which is more favorable for the organism. Getting it? Now, tall height
Okay.
is always favorable over short height. The
Short
plant
height, yeah.
is getting benefited more by tall, no?
Mm-mm.
So that's a dominant one.
Okay, got it.
Are you getting it?
Getting it.
So between tall and dwarf, tall is dominant over dwarf. And a
Yeah.
dominant allele has got a capacity to suppress the effect of another allele, which is called recessive allele.
Correct. You told me, yes.
So recessive means what? It is not expressed in the presence of an alternative allele. कि अगर उसके सामने कोई दूसरा डोमिनेंट एलील होगा तो ये शांत बैठेगा, नहीं अपना मुंह नहीं खोलेगा.
Correct. Yes.
It is recessive allele.
Yes. Yes.
Okay. Recessive allele is expressed only when there is another recessive allele.
Oh, okay, got it.
Otherwise, the recessive allele will not express.
Okay, got it. Yeah.
That leads to two more words, something called homozygous and heterozygous.
Homologous. Oh,
Homologous नहीं, वो अलग है. This is homozygous. Homozygous and heterozygous.
Heterozygous.
Okay. Now see, whenever you're telling or giving an idea about the genetic composition of that character, you're mentioning the alleles. So I write it like, suppose I write capital T, capital T.
Yes.
Capital T stands for tall height.
Tall height.
So this way, when I write both the alleles, so both of them are the alleles for tallness, that is dominant.
Correct.
This way, when you write, it is called the genotype. The genetic constitution. That means this is giving me an information that inside the cell, the two genes or the two alleles are there for tallness.
Okay,
तो ऐसे जब हम लिखते हैं तो उसको जीनोटाइप करके बोलते हैं. It's like on paper you write with the help of letters. So mentioning the genetic constitution with the help of these letters.
Okay.
Okay. So when the genotype is written in such a way that both the dominant alleles or both the recessive alleles.
Yes.
Together, that means both the alleles are either dominant or recessive. Then we call it homozygous.
Okay. Yes.
Identical.
Sorry, what is homozygous?
When in a genotype that is written, both the alleles are identical, both dominant or both recessive. Then it is called homozygous.
Okay, got it.
Also called pure genotype.
Pure genotype.
Because kind of impurity नहीं है ना, उसमें दोनों same है.
हाँ, correct.
Getting it?
Yes.
Okay, so tall अगर है तो capital T, dwarf है तो small t. So while writing the genotype, we always use the first letter of the dominant trait. Tall is dominant over dwarf. So we always use the letter T.
T.
And dwarf के लिए हम लोग D नहीं लेते. We write small t.
Yes.
Getting it? So letters remain the same. एक capital, एक small.
Okay,
capital indicates dominance, small indicates recessiveness. Now there can be a condition where the one allele is dominant, other one is the recessive one together.
Yes.
This can also be possible. उस condition को बोलते हैं heterozygous.
Heterozygous.
When there are contrasting alleles. Getting it?
Yes.
Now when you write the genotype like this, capital T, small t. So which one will be expressed?
Both.
Capital T, small t, both will be expressed. One is dominant, one is recessive.
Yes.
तो recessive express होगा dominant के सामने.
नो, उसके लिए एक और एक रिसेसिव चाहिए. तो डोमिनेंट टी, बड़ा टी.
कैपिटल टी विल बी द वन विच इज एक्सप्रेस्ड, उसी को डोमिनेंट बोलते हैं ना.
यस.
आर यू गेटिंग इट?
गेटिंग इट,
Whenever
गेटिंग
there
इट.
is one dominant and one recessive, it is only the dominant which is expressed and not the recessive.
Not the recessive.
उसको heterozygous बोलते हैं.
Yes, got
Getting
it.
it? So understood the meaning of the word phenotype, sorry, genotype properly?
Yes.
Sure.
Sure.
Genetic constitution or the makeup. Now, whenever there is a particular genotype that is written, based on that, some character is expressed, some trait is expressed.
Like?
जैसे capital T अगर लिख रहा हूँ, so how will the organism be? Tall or dwarf?
Tall.
So that expressed character is called phenotype.
Okay, got it.
So for any particular trait, the external appearance of that individual is called its phenotype.
Okay, got it.
Round seed is a phenotype. Purple flower is a phenotype. Okay, constricted pod is a phenotype. Any dominant or a recessive character which is expressed in that organism, that is called as phenotype.
Okay,
And
got it.
every phenotype has got one or more genotypes. Every phenotype has got one or more genotypes. See here.
So wait, I'm confused
Yes.
between.
I'm confusing you. See. Now suppose there is an organism that you see which is tall.
Yes.
You can see that the organism is tall.
Tall.
So looking at the organism, you say कि ये tall है. जैसे मेंडल ने pea plant को देखा. Okay, it is tall.
Yes.
But looking at the organism, you cannot say whether the tallness is because of homozygous condition or because of heterozygous. Both the conditions can give tallness.
Yes, correct.
Are you getting it?
Oh, yes.
So that is inside the cell. वो उसके genes में ना, उसके alleles में. So this is your genotype. Getting it? And this is the phenotype.
Yes.
Now have you understood the difference? Getting it? Now if I say that an organism you see is dwarf.
Yes.
Okay, छोटा है, short है. So if I ask you if dwarf is the phenotype, what is the genotype of the organism then? Think and answer. What is the genotype of organism which is dwarf?
Small t.
एक ही लेटर नहीं होगा ना genotype में.
हाँ, तो I said small t.
Small t. ऐसे.
Yes.
So that is the correct. That is the genotype. Because capital T अगर एक भी आता है तो dwarf नहीं रहे.
So it is not dwarf. Yes, correct.
Okay. So this way the question is asked. This way the question can be asked. If this is the phenotype, what is the genotype?
Okay, easy.
Now understood. If the question is like, if the genotype of an organism is capital T, small t, what is the phenotype?
Capital T, small t.
Phenotype क्या होगा?
Oh, phenotype. T. Phenotype will be.
Phenotype means you will tell the character. If the genotype is capital T, small t, I am asking you what is the phenotype. So what will you say?
Dominant and recessive.
Supriya, I am asking, concentrate. If the genotype of an organism is capital T, small t, what is the phenotype of that organism?
Tall.
Tall. Only tall. Tall and dwarf दोनों रह सकते हैं क्या? ऐसा फ्लक्चुएट होता रहेगा. Tall, dwarf.
I got it. Yes, I got it.
Are you getting it?
Yes, I am getting it.
Concentrate, understand. No confusion.
Yes.
Getting it? अगर
Getting
पूछा किसी भी ऑर्गेनिज्म के लिए कि व्हाट इज फिनोटाइप, तो यू आर गोइंग टू डिस्क्राइब अ कैरेक्टर, वहां पे लेटर नहीं बोलना है,
Okay,
वहां
got
पे
it.
जीन एलील नहीं बोलना है, कैरेक्टर बताना है.
Yes, got it.
आर यू गेटिंग इट?
Getting it.
ठीक है. एंड कैरेक्टर अगर पूछा कि फिनोटाइप है एंड उसका जीनोटाइप क्या है, देन यू विल टेल द लेटर.
Yes, of course. Yes.
गेटिंग इट? ऑफ कोर्स
Got
बोल
it.
रही है बट टी कर रही थी
Yes.
वहां पे. एंड अ पर्टिकुलर ऑर्गेनिज्म कैन नॉट हैव
Get
टू
it.
ट्रेट्स फॉर द सेम
समझा.
कैरेक्टर. जैसे हमने
Yes.
कलर ऑफ द आईज देखा, ब्लैक, ब्लू, ग्रीन एंड ऑल. कैन अ सिंगल पर्सन हैव टू कलर्स इन बोथ द आईज? एक ब्लैक है, एक ग्रीन.
हाँ. Brownish black.
एक आंख ब्लैक है,
Yes.
एक ब्राउन है, ऐसा होता है क्या?
No. नहीं.
गेटिंग
नहीं.
इट? कैन अ पर्सन हैव मोर देन वन ब्लड ग्रुप?
No.
No, because it is fixed means it is fixed in every cell of the body.
Yes.
Are you getting it?
Yes.
ठीक
Getting
है,
it.
ये सब पॉइंट्स अच्छे से समझ जाना है.
Yes.
ठीक है. तो वी हैव
ठीक है.
डिस्कस्ड अबाउट फिनोटाइप, जीनोटाइप, होमोजाइगस, हेटेरोजाइगस.
Heterozygous.
एंड
Yes.
हेटेरोजाइगस अगर इंडिविजुअल है, जैसे ये कैपिटल टी स्मॉल टी बोला, उनको
Yes.
हाइब्रिड भी बोलते हैं. सच एन इंडिविजुअल इज आल्सो कॉल्ड
Oh yes.
हाइब्रिड.
Hybrid. Yes.
हाइब्रिड का मतलब कॉन्ट्रास्टिंग हो गया ना, एकदम
दोनों का
ऑपोजिट, टॉल,
mix.
ड्वार्फ,
Yeah.
कॉन्ट्रास्ट.
Correct.
मिक्स नहीं,
Yes.
कॉम्बिनेशन ऑफ बोथ.
Combination. Yes. I was
इसलिए
going
इसको
to say that.
हाइब्रिड. Got it
Yes.
now? Okay.
Got it.
What
Got
is the meaning of F1 generation?
I know. I know. I know. ये मैंने पढ़ा था. ये मैंने पढ़ा. F1 generation. Wait. Inheritance. No.
F1 generation means what?
First something generation.
First something,
What?
first filial.
Filial. Yes, correct.
First filial generation. Okay, first
Yes.
generation, wherever we start with the inheritance, that is called as the parental generation.
Yes.
And when the gametes are produced, the fertilization takes place and the new generation is produced. That is called
Yes.
F1 generation.
Yes, got it.
Okay, now F1 generation can also become the parents for the next generation.
Yes.
तो F1 generation का जब अगला generation आएगा, we call it F2 generation.
Okay. Oh,
F2
okay,
का जब
got it.
अगला generation आएगा तो F3. Like that we can keep on telling.
Yes, got it.
Okay.
Yes.
Now
Yes.
there is something called pure line.
Pure line.
Pure line करके एक word है. Okay. See, I'll show you this way. There is a parental generation having.
Pure line वो है ना जो first and then उसके बाद से.
Listen. There is a parental generation.
Yes.
Capital T. That means homozygous tall.
Yes.
Homozygous tall. Crossed with another homozygous tall. Whenever you're writing a cross or representing it, this is how it is shown.
Okay, got it.
इसको बोलते हैं क्रॉस.
Yes.
ये लिखा हुआ ऐसे रिप्रेजेंट करते हैं, बट प्रैक्टिकली इट इज डन इन अनदर वे.
Okay, got it.
So it's like parental generation are dominant homozygous tall crossed with another individual which is also homozygous tall. दोनों homozygous है same character के लिए. Now this is the genotype that is written, consisting of 2 letters.
Correct.
2 alleles.
Yes.
Okay. When these, from this genotype, when the gametes are produced, gamete production mein it is becoming half. You remember meiosis?
Oh, gamete. Yes, of course.
So this
I
will
remember.
produce 2 gametes. 2 gametes. First one will have one of the T. Second one will have the other T. That means 2 gametes are produced, but there are only one type of gamete that is produced. Gamete containing only capital T. अभी ये देख, suppose it is heterozygous tall. ऐसे.
Yes.
तो इसमें से जब गैमेट बनेगा, So there will be two types of gametes. One of the gamete will have capital T,
capital T and
other
small
one
t.
will have small t. So ये two types हो गया because दोनों में
got it.
अलग है.
Yes.
But here दोनों या 100 गैमेट भी बनेंगे तो भी सब एक ही टाइप के, सब में कैपिटल टी.
Yes.
अगर यहाँ पे 100 गैमेट्स बन रहे हैं तो 50 में कैपिटल टी है, 50 में स्मॉल टी.
करेक्ट. Yes.
इसको बोलते हैं सेग्रीगेशन, सेपरेशन. Getting it? Now here, come back to this. So इसमें भी एक ही टाइप का गैमेट बन रहे हैं.
Yes.
Getting it? So whenever during fertilization, the gametes of one parent fuses with the gametes of the other parent, right? So the gamete of one parent always fuses with the gamete of another parent, okay? And the offspring that is produced will have a genes from both the parents, okay? This offspring that is denoted like this, as I mentioned earlier, it is called as the F1 generation, okay? Now this F1 generation can also become a parent for another generation. And suppose if this one is also crossed with another homozygous dominant condition, same as that of the parental generation, same way the cross will be done. There will be fusion of the gametes, and the F2 generation also which you get will also be homozygous dominant. Okay, so it's like if suppose this kind of cross is continued like this from F1, F2, F3, F4, like for a series of generations, and across all the generations you are seeing that there is no mixing up of characters of any recessive trait. It's only the same characters getting repeated. Then we call it as a pure line. Okay, so it's a group of individuals which is homozygous or true breeding for one or more traits. Here I give the example for dominant, that is tallness. The same thing can happen even for dwarfness. Getting it? So there is self-fertilization that can just continue, which is called as pure line. If at all there is one individual also coming in between, which has got a recessive trait or a heterozygous condition, then it doesn't remain a pure line. That's how you have to understand it. Okay. Then there is something called monohybrid. Okay, monohybrid means, so we have already seen what is the meaning of the word hybrid, a heterozygous condition. Okay, now it's like suppose I show you this way. If there is one parent which is homozygous tall, we generally take the example of tall and dwarf for a better understanding. So suppose if it is homozygous tall crossed with another parent which is dwarf, that means both the parents are homozygous for contrasting characters. Okay, so here this parent will produce a one type of gamete, all of them will be capital T. This parent will produce again only one type of gamete, all of them is small t. One type. It can be thousands of gametes also, but they are of one type. Okay, so when these two gametes will fuse, the F1 generation will turn out to be heterozygous. It will be heterozygous. Okay, so here we have considered only one character, that is height of the individual, two traits, tall and dwarf, and we have crossed. So you have gotten hybrid offspring. Such a cross or such a organism is called a monohybrid. Mono means single. That means it is heterozygous for a single pair of alleles. Then it is called monohybrid. Okay. Same way, there can be something called dihybrid. Now, when you say dihybrid, the word di indicates two. Okay. So it's like here in this chart, you can see this is a classical example of a monohybrid cross, which Mendel has done. A tall pea plant crossed with a dwarf plant. Now, whenever you are talking about the phenotype of a recessive condition, we don't generally use the word dominant or recessive or homozygous and heterozygous. Like we don't say homozygous dwarf because we don't need to. Dwarf is always homozygous. Okay, dwarf can never be heterozygous. But for the dominant phenotype, we always mention the word, whether it is homozygous dominant or heterozygous dominant, because without mentioning, it is not possible to tell. That is something you have to remember. So the phenotype is tall and dwarf. Genotype we are considered. If the dominant phenotype is written without homozygous or heterozygous, then it is understood it is homozygous. Okay, so gametes capital T small t, you get heterozygous individual. And now this heterozygous F1 which you have got, that is capital T small t, that is undergoing something called selfing. Selfing means crossing the F1 individual with the same F1 individual. Same means similar type. And here they are becoming the parents now. So because it is heterozygous, there are two types of gametes produced, capital T, small t. So there are two types of male gametes, two types of female gametes. So that's why when you represent it in the form of a chart, it is called as a Punnett square or a checkerboard method. Okay, any gamete of the female can fuse with any gamete of the male and you will get a respective offspring. So here, for example, you will get capital T, capital T. That is homozygous term. Here you will get capital T and a small t. That is heterozygous. Okay, here you will get small t, that is dwarf. Like that, you prepare a Punnett square and you fill up the possible offsprings. Okay, so Punnett square is a easier method to understand what type of offsprings are obtained and in how many numbers. Because from that, you will be able to calculate something called the phenotypic ratio and the genotypic ratio. Okay, it's like, for example, over here, homozygous tall. Okay, phenotypic first. Phenotype means the external visible appearance. So in this cross, how many tall to how many dwarf? So you are getting 1, 2, 3 tall is to 1 dwarf. Because here homozygous tall, this is heterozygous tall, this is heterozygous tall, and here it is dwarf. So you are getting a phenotypic ratio of a monohybrid cross as 3:1. 3 tall is to 1 dwarf. Okay, which you get in the F2 generation. Similarly, if you see the genotypic ratio based on the genotype, so you are getting one which is homozygous tall, 2 of them are heterozygous tall, and 1 dwarf. Okay, so here see 1:2:1 is the genotypic ratio of a monohybrid cross. Okay, so that's where you can understand phenotypic ratio and genotypic ratio. Okay, regarding say homologous chromosome, we have already seen. Okay, identical chromosomes having genes in the same location. We have already seen F1 and F2 generations. Progeny means the young ones. Okay, that's the word we use in genetics. Selfing is also called inbreeding. Okay, that means crossing with the same generation of springs. That's called selfing. Okay, back cross, test cross, and all we will see together when we will discuss about Mendelian experiments. Okay, in detail, we will see how Mendel carried out the experiments, monohybrid cross as well as dihybrid cross especially. And we will see how to calculate the Punnett square, the ratios. And from that, we will understand what is the actual meaning of back cross and test cross. Okay, and what is the significance. And based on the experiments which are carried out by Mendel, we will see the laws of inheritance which was put forward by Mendel and how he explained that. That will complete the Mendelian part of the genetics. Okay, and because Mendel could understand only this much based on the experiments that we carried out. There are a lot of other things also in genetics which Mendel had never observed or come across. Okay, so that comes under something called neo-Mendelian genetics. We are going to see the different methods of inheritance in different other organisms also, which Mendel could never discover. That will be the other further part of the topic. Very, very interesting aspects are there. Lot of crosses, and how to cross them, how to solve them, everything will be discussed. Okay, so hope everything is clear till now. These terms that we have discussed, the terminologies, the genetic terminologies are the most important part, but that becomes the base for understanding this topic. So please make sure that you are revising these terms really well, understanding them, because this will come again and again in the further part of the lesson. Only then you will understand it better. Okay, so we will stop here for now, and Mendel's inheritance and the neo-Mendelian genetics we will continue in the next session. Okay, thank you.